Okay, before we get started, if you've not written out your hypothesis and your experimental design, or at least an idea, stop and do that now. You need to answer these questions first before we begin. That's part of this lab and part of the skill of being a laboratory scientist. You need to know how to think about what you're trying to test, create a hypothesis, and design an experiment. So pause now. Okay, I'm assuming that you continued because you've already done that. What I'm going to do now is show you what I did to come up with an experiment, and we're going to kind of work through this. You may have had a different idea, that's okay, but kind of stop this. You may want to pause here and kind of look at what I did. What I've done is jot down the indicators that we have. I didn't put down what they test for because that's something that you should be able to figure out. I've also sketched what I think the experiment should look like, maybe a beaker with the dialysis tubing and two different solutions, one inside the tubing, one outside. And then I've written down, if we have 4 different indicators, we could test the liquid in the beaker and the liquid in the dialysis tubing bag with all 4 indicators to see what we get, and do that before we let it sit for a while. And then, after it's had a chance to do some diffusing, we'll come back and do it again, basically run the exact same 8 tubes and see if any of that changes. Okay, as we get started, there's a lot here. We've got lots of different things to look at. Notice that I've already made my beakers of 2 different solutions, and I've mixed 2 of the chemicals together so that we can kind of combine this and not do each one individually. I have my dialysis tubing. Looks like tape. It's not sticky though, and it's actually a tube once you soak it. I've got a couple of pieces already cut in this beaker. They've been soaking so that makes it easier to open them up, and when you rub them between your fingers, you can see that they actually become a tube rather than just a strip. And this is a semi-permeable membrane, and we'll see what things can pass through it and which things can't. Now the blue things are clips that will go on the ends of the dialysis tubing to basically seal the ends off. Okay, I've got my solutions, I've got my test tubes, I have my scale, we'll talk about that in a minute, I have my indicators here, and let's kind of run through some of these real quick. My solutions came from powder, so here's my albumin, came from egg white. Albumin is a protein. My cornstarch, we've used starch before. That's carbohydrate, complex carbohydrate. There's the sodium sulfate, a salt, and a sodium chloride, another salt. And again, those were mixed ahead of time and are already in the labeled beakers. Okay, here are our indicators. We'll run through those. You've already seen iodine before, and hopefully you remember what it tests for. There is our barium chloride. It's a little blurry there, but that's barium chloride in a dropper bottle. This is silver nitrate. Silver nitrate is also in a little dropper bottle. And then these are the albusticks or test strips, and you can see that there are a bunch of little squares. Those are actually on the test strip, and we're only going to be using one of those squares. It's the 4th one from the end, and it tests for protein, and it will change color if protein is present. We'll see a better look at that a little bit later on. And you can see that we can just compare those little squares. You can actually use that to check all kinds of things. Alright, so now that we've done that, we're ready to make our dialysis tubing bag. And so I'm going to go ahead and put inside the bag the solution that we're going to test there. And if you remember my design, we were going to put the albumin NaCl mixture in the bag and the sodium sulfate and starch mixture in the beaker. You could do it the other way around, it doesn't really matter, and you could do this experiment lots of different ways, but that's how we're going to do this one just to see what happens. Okay, we've clipped our dialysis tubing on one end. It looks kind of like a strip of plastic, but if I rub it, I can open it up, and then we're going to use a transfer pipette to put some of the albumin NaCl mixture into the bag. So let's see if we can get it in there. It's a little tricky because that bag is kind of hard to separate until you get it started. Okay, we'll squirt a little bit of that in there, and that kind of helps open it up a little bit. Dialysis tubing is not exactly the same as a cell membrane, but it makes a good comparison in this lab. Cell membranes are also semi-permeable. Certain things will pass through a cell membrane, certain things will not. Dialysis tubing is not alive, it's not from a cell, but it is also semi-permeable. Remember, our goal is to find out which compounds, albumin, NaCl, Na2SO4, or starch, or water, will pass through the bag. Now, we haven't talked about this yet, but how are we going to find out if water goes in or out of the bag during our experiment? Is there a way that we could measure whether or not water goes in and out? Think about that for a few minutes, and then we'll come back to that question. Okay, I've almost got it filled up. You can see there it's kind of tricky to get that liquid in, and I'm dribbling a little bit as I'm trying to get it into that opening at the top of the bag. It's almost full. Okay, now we're ready to clamp off the other side. Okay, you can see it's pretty plump and holding our albumin NaCl mixture. Kind of dab off the extra liquid there, and we'll clamp off the other end of the tube. And you can see it just kind of hinges open, and then we need to make sure we squeeze it really tight to seal that bag. And then we're going to need to rinse it off so that we don't contaminate our beaker with any of the solution on the outside of the bag, because we were kind of messy there. So we'll kind of clean that off a little bit. And I'll just use a little bit of the distilled water and just rinse it. Okay, if you remember, we needed to find out a way to tell if water will diffuse into or out of the bag during our experiment. And there's our bag, it's all ready to go. Now if we want to find out if water moves through the bag, we can just stick it on the scale and get its mass now, and then compare that to the mass at the end of the experiment. Okay, its original mass is 21.15 grams, 21.15 grams. Okay, and now we're ready to put our dialysis tubing bag into the beaker, and we'll cover it up with the sodium sulfate and starch solution and let it sit for a while so that the materials that can diffuse and water, if water does as well, we'll see if those materials will diffuse through the bag. And there we go. We've got our dialysis tubing inside our beaker, and the solutions are separated by the membrane. Okay, now we need to take a look at our test tubes. I've got them labeled 1 through 8, just as I had written on the whiteboard at the beginning. And what we're going to do is go ahead and run our indicators on these solutions. And we know what we put in the bag, we know what we put in the beaker, so we can just take these same solutions from our original source and run our indicators on those directly. So what I'm going to do is just put a little bit of those solutions. We're doing the sodium sulfate and starch in tubes 1, 2, 3, and 4. So I'm just putting a few milliliters in there. Again, the volume doesn't really matter very much because all we're looking for is the result of whether or not an indicator changes color. But it's about 3 milliliters in each of those 3 tubes. Okay, and test tubes 5, 6, 7, and 8, we're going to do the same thing, but we're going to use the albumin NaCl solution. Again, putting about 3 mils in each tube. And then we'll test each of these with our indicators to see what we get. And that'll help us practice a little bit with these and verify our starting point for our experiment. Okay, there are our tubes. They've got our solutions in them, and we're ready to go. Alright, so our first tube we're going to test with iodine. We're going to do that for test tube 1 and test tube 5. You may want to follow along in the data section of your lab manual where you can write down what we're doing and record the results. Okay, and so I've just added a couple of drops to each of those. We don't have to do anything special. All we need to do is add them and look. Okay, we've used iodine before, so hopefully you can tell that number 1 gave a positive result because it turned black, and number 5 did not because it turned orange, which is the color of the iodine. Okay, let's go to barium chloride, and we're going to add barium chloride to test tube 2 and 6. And this one's a little bit trickier to use because the starch solution is already cloudy. What we're looking for is a white precipitate to form. If you remember what barium chloride tests for, it tests for sulfate. If we mix these together, you should be able to see that it's kind of hard to tell, but test tube number 2 became a little bit whiter, a little bit more opaque. It's hard to see in the video, but it did become more opaque. There was a white precipitate. Alright, now we're going to switch to the silver nitrate, AgNO3. We'll add a couple of drops of that to each of the test tubes 3 and 7. Okay, we added a few drops in there and didn't really see much of a change. We'll add a few drops to that one. Oh, look at that. So as soon as we added the drops, it turned as cloudy as milk. And that was the white precipitate that forms because silver nitrate tests for chloride, and test tube number 7 contained sodium chloride. Okay, I've got my 2 test strips set out, and this is the key to what those test strips actually will test for. And I apologize that it's blurry. The print is very small, it's hard to see. But we're going to be looking at just one of those squares on the test strip and comparing it to the colors that are on the bottle. And the way that you do this, you hold the handle of the test strip down at the bottom of the, or the kind of the bottom of the bottle. You line up the squares, and then you can see there that they match color. If any of those colors change, for example, if we see green in the 4th square, that 4th square starts off as yellow. If it becomes green, that is a positive indication for protein. So this is our way of testing for the presence of protein. We're going to do that for test tubes 4 and 8. And we already know that test tubes 1 through 4 have starch and sodium sulfate, so they do not have protein. We should not see a color change. Test tube number 8, however, 5 through 8, did have protein. So we should be able to see in test tube number 8 a difference. Alright, so let's compare. If we look at our 4th square from the right side, you can see there that it's still yellow. Okay, let's try the same thing with our test tube number 8. Test tube number 8 contains albumin. Albumin is egg white protein. So if you eat an egg, you're eating albumin. It's also a very common protein in us. Okay, and it does take a few minutes for it to really change color, but you can see there, hopefully, that it's already starting to turn green. But we'll have to give it a couple more minutes before we can see a real obvious difference. But it does have a little bit of a greenish color already. So let's give it a couple of minutes. We'll come back and look at it again. Okay, with the magic of film editing, we're now ready to look at it. You can see there on the on your left, that one is the first one we did, and it is more yellow. The one on the right is the second one that we did, and it is more green. And you can see the difference there fairly well when you just compare the 2 strips side by side. Okay, we're ready to let this our dialysis tubing sit for a while, and we're going to come back and see if any of these results change after this has had a chance to sit. Okay, so it has been about 45 minutes. I've washed the test tubes, and they're ready to go. And you can see that the dialysis tubing has just been sitting in the beaker for about 45 minutes now. Before we do anything else, we want to weigh it. Again, if the mass goes up, that means water diffused into the bag, and if the mass goes down, water has diffused out of the bag by osmosis. Some students have asked in the past if the movement of solute molecules would affect the mass, but it really won't. But the movement of water will. All right, it looks like our mass is 22.19 grams. 22.19. Might be 22.20, kind of wavering there, but we can go with 22.19. Okay, so you'll need to compare that to our initial mass and determine for yourself what happened. Did the mass go up? Did it go down? And what does that mean? Okay, and I'm just getting our test tubes ready so that we can run the exact same procedure we did at the beginning. We're going to take some liquid from the bag itself and liquid from the beaker, and we're going to put those in the test tubes. We'll put the liquid from the bag, albumin and NaCl mixture, in test tubes 5, 6, 7, and 8. We'll put the liquid from the beaker into test tubes 1, 2, 3, and 4. Okay, all I did was unclip one end of the bag, and we're just going to use the transfer pipette. This is a clean one, and we're going to extract some of that liquid and put a little bit in each test tube. Again, it doesn't really matter how much because we're just looking for an indicator to show us if anything has changed. Okay, so I'm just going to squirt a little bit in each one of these test tubes. Again, this is from the bag to tubes 5, 6, 7, and 8. We already know that the bag contained albumin and sodium chloride because that's what we put in there to begin with. What we're now looking for is if anything new shows up. Okay, and there's our last test tube. We're done with the dialysis tubing bag. We can set that aside. We have our samples from the bag now. I need a clean transfer pipette, and we'll fill tubes 1, 2, 3, and 4 with the solution in the beaker. And if anything from the bag has escaped the bag and diffused out into the beaker, we should be able to find out with our indicators. So again, all we need is a little squirt into each of the test tubes, and we're going to run the same tests, see if anything changes, and we're ready to go. Okay, let's start with the same ones we did last time. We'll go in the same order. So that means we want to start with iodine, and there's the iodine. And we'll just add a couple of drops to test tube 1 and 5, just like we did last time. Make sure you record your data. Test tube 1 turned black, test tube 5 turned orange. What does that mean? Be able to interpret your results. Okay, our next one, barium chloride. Put a few drops in 2 and 6 in test tube number 2. Kind of hard to see what's going on there, but I don't know if you can tell with my white lab coat, but there we go. You can see it's starting to turn white, so we do have a positive result. It's not a real strong positive result though, but we do have a positive result. And if we add it to test tube number 6, we definitely get a positive result. So let's say for both of those we did get white, but 2 is just kind of weak for some reason. Alright, let's go to our silver nitrate, AgNO3. We're going to add that to test tubes 3 and 7. And in test tube 3 we definitely got white precipitate, looks like milk. And in test tube 7 we also got a white precipitate. Again, looks like milk. So positive results for both of those. Alright, in the last one we're going to test for the presence of protein with the albusticks again. And again, these have lots of little test squares on them. We only really just need to look at one particular square. And so I'm going to stick it down in there. Again, all we're trying to do is get that fourth square from the end wet. So that's what I'm trying to do here. I'm just tilting the tube and trying to get it to the point where it moistens that fourth square. And we'll see if we get a color change. Okay, and on test tube number 4, you can see that the fourth square from the end still looks pretty yellow. Let's try test tube number 8. Again, we just need to get the end of it wet, enough to get that fourth square. Okay, and you can see there, it still looks pretty yellow at this point, but we know that there should be some protein present, so we'll give it a couple of minutes to sit, and then we'll come back and look at it because it does take a little time. So you should have some results. We'll double check our albusticks results, but write down this data and then make sure you understand it, because having data without really understanding what it means is a waste of time. All right, we'll be back in a minute. And we're back. It's been about 5 minutes, and we can now take a look at these strips and look for a change. The strip that was on the front was from test tube 4, and that still looks pretty yellow. The test strip from the back, from test tube number 8, it looks green. It's kind of blurry, it's hard to see there, but I think you can see the color difference fairly well. Alright, at this point you have the data, so it's now up to you to interpret what your test tubes told you and figure out what all of this means in terms of diffusion of materials.